Thank you, Simon Foundation. Thank you, everybody, for being here, right? So we decided the title of our talk today is Tales of Diverse Hearing and Sound Localization, Specialty from Owls to Humans. So I'm proud to show a photograph of my group. Fortunately, very happy diversity, right? Separate races and genders. And also we have collaborators all across this country and in many different countries. Unfortunately, like shown here, we are funded by several NIH programs, including Brain Initiative and Collaborative Research. So based on the title of the talk that was decided today, this would be the items we are going to talk about our schedule today. Brain function towards hearing and sound localization specialties and from owls to human. So the first point, brain function towards hearing and sound localization specialties, right? So hearing is a very important function across species, right? We all hear things, and all across animals, right? Frogs call each other, birds communicate, right? Bats use hearing to localize sound. Owls, yeah, our special barn owls, they use hearing to find food at night, and also we use hearing for many things to communicate each other, right? And even insects hear sounds like this. Some flies, they detect crickets to lay eggs on them when they're calling. So hearing is a very important function across nature. And for humans, hearing has been fascinated, and a lot of art has created, a lot of literature and art about hearing, sculptures about hearing, and books about hearing, and symphonies, and also music, right? Which is sound that we have created. We enjoy life, and maybe across the universe by creating music, right? Life spins across the universe, we are creating new sounds, right? That could be appreciated across the universe. So then hearing in nature is different, how different species hear sounds, right? So this is our ear, right? The Pena is our ears, and middle ear and inner ear. Birds have a slightly different shapes of the ears. Insects, even here, with their antennas, right? And even fish hear sounds, and even plants are supposed to hear, right? With the vibration of sounds on the leaves, right? So the hearing is general across nature, right? And our ears are different across species, right? This is the shape of our ears, our Pena, and the other species have different ears. Some other species have movable ears, right? And birds, like owls, they have different shapes of the ears, right? They don't have Pena like we have, because they have feathers, right? So there are differences of ears across species, and also there's a difference of ranges of frequencies we can hear, right? These are all the frequency ranges all across species, right? There are some species that can only hear narrow bands of frequencies, right? Some species hear high frequencies, right? Like bats, for example. Bats, whales, dolphins, they can hear high frequencies, right? And we have a certain range of frequencies that we can hear. And how our ears detect sounds of different frequencies has been investigated, right? And what was found by George von Beckersey, right, who won a Nobel Prize, right, 1961, discovered how the ear detects different frequencies, and it was found that actually there is a traveling wave. There's a basilar membrane in the ear that vibrates at different locations for different frequencies, and this is how ears detect different frequencies. And so George von Beckersey won a Nobel Prize on physiology or medicine, actually. And actually, it was a very important discovery for medicine, because knowing how our ears work helped a discovery of generating an implant, a cochlear implant, which is used for people that have hearing loss at different ages, or even for babies that cannot hear. It was discovered that because the ears detect frequencies in different locations of the ears, or detect the frequencies, if an electrode sounds at different frequencies that are exciting the cochlea at different locations, it could be induced hearing, and that was found by creating the cochlear implant. So it was an important discovery for health and for medicine to understand how we hear. So one important thing is localizing the location of stimuli by our brains, all across sensory modalities, right? When we see things, we have to localize visual stimuli, even in our body when we are touched, right? We localize where the stimulus, what part of our body the stimulus is. But also sounds, we have to localize sounds. However, localizing stimuli varies across sensory modalities. And for example, in the visual system, with our eyes, with the retina, we detect visual stimuli, and the location, right? If this is what we are seeing, right? The location of visual stimuli determines the location in the retina. However, the location of sounds is not directly related to the location of activity in the ear, because the location in the ear is for different frequencies, but not location. Therefore, sound localization, localizing direction of sound, has to be computed by the brain, unlike what is the perception of visual location and touching, which is automatic. So for localizing sounds, the brain has to compute information. And the cues that are used for sound localization are monaural cues and binaural cues. Right? So we'll go there rapidly. Monaural cues are information from single ears, right? Our single ears provide information that gives us where a sound is. And these monaural cues have been, for example, investigated in humans. These are our ears, and on the right is a plot of what are the power of different frequencies when sounds are different locations, and specifically what was found in our ears, our hearing, is that the pattern, the power of different frequencies varies, right? And this is for a sound 30 degrees below the horizontal line, 0 degrees at the horizontal line, 30 degrees above. And you see that there are different curves. Means that the pattern of frequency changes for sounds at different elevations, right? And it has been shown and investigated that in humans, the shape of the ears, right, when it's changed by inserting molds, the pattern, the power of frequency changes, right? And what it was found on human volunteers, they have these molds for several days. As soon as they get these molds, right, they no longer are able to localize, determine the elevation of sounds. But then with a few days with these molds, they learn how to reuse their ears, and they recover the ability to localize sounds in elevation. And when the molds are removed and they use their previous ears, they immediately are able to localize the elevation of sounds. So these are monaural cues of sound localization. The other thing I mentioned is binaural cues. Binaural cues is comparing information between the ears, which is also used for sound localization. And specifically, these binaural cues are called interoral time differences and interoral level differences. Interoral time differences, difference in time of sounds that arrive to the ears, with the left and right ears. Also differences in intensity of the sounds that arrive to the ears. So specifically, these interoral time differences in humans, right? The sounds arrive at the same time to both ears when the sound is in the front, and they are around different times when the sound is in the periphery, right? And given the size of our head, right, the distance between the ears, the maximal difference in time when the sounds are in the periphery is around 0.6 milliseconds for humans. Still a very small difference in time that our brain can detect. And it was also investigated in humans, and it was found for different frequencies of sounds, right, how much a sound can be moved based on detecting interoral time difference. So for higher frequencies, has to be very, very large changes, right? For high frequencies, our ability to detect interval time differences is very low, right? This is an example of 2,000 hertz. For lower frequencies, right, there's a better discrimination of interval time differences, right? Even though in the front of the head, the changes are more selective, there's more discriminability in the front than in the back of the head or the periphery, based on detecting interval time differences, right? So basically, we humans can detect interval time differences are at low frequencies. This was one of the binaural cues. The other cue is interval level difference. And what was found in our head, right, is that the interval level differences changes most across different locations for high frequency sounds, right? And this is because of acoustic properties. Basically, low frequencies are known to be able to go through walls and doors, right, than high frequencies. And that's why the difference in intensity in the sounds between the ears is larger for high frequencies, because our head can create the shadow of the intensity for higher frequency sounds. So then there is a difference between the frequencies that humans we can detect interval time and level differences, and it was a theory of human sound localization called duplex theory of human sound localization. Based on this discovery, this theory says that we use interval time difference in green, the green plot, for lower frequencies, and we detect interval level differences for high frequencies, right? So these binaural cues are an important cues that the brain uses to compute the location of sounds. And the brain has to detect differences in times and difference in intensity. And basically, difference in time are detected by nuclei in the brainstem. Specifically, medial superior olive is a nucleus in the brainstem in mammals and humans that detects interval time differences. Lateral superior olive detects interval level differences. In birds, these regions that detect interval time difference and level difference, they have different names, right? The nucleus laminaris is the region that detects, that's equivalent to the medial superior olive in mammals, and lateral lemniscus is the region that detects interval level differences in birds. So they have different names. As a research, because owls are very specialized sound localizers, the barn owls, because there are many different owls, but barn owls are nocturnal, and they are very specialized in finding food at night by detecting sounds, detecting prey, right? And this inspired research of sound localization, and my former mentor at Caltech, Marco Niche, discovered a map of auditory space in the midbrain of the owl, what was considered a seminal discovery of how our brain encodes and computes sound localization. And this induced decades of research in owls to investigate how these maps are created, right? So specifically, it was found that owls, unlike humans, owls have asymmetric ears, and they use interval level differences for determining the elevation, the position in elevation of sounds, because they have asymmetric ears, and when a sound is below the horizontal level, it's louder on the left side. When it's above, it's louder on the right side. So owls use interval level difference for determining the elevation of sounds, and interval time differences for determining the horizontal position of sounds, right? Different from us, that we use interval level difference and interval time difference for horizontal positions, right? And then this is external nucleus of the inferior colliculus. This is where the first map was found. However, this map projects to another map called optic tectum in owls too. There's another map, and this is equivalent actually to the superior colliculus in mammals, right? And there has been a lot of research about to figure out how this map of auditory space is created in the owl. So the specialized behavior of sound localization of owls have been also used to test models of sound localization. For example, there was an engineer, Jeffrey's model, an engineer who imagined how our brain, right, can detect differences in time. It was very fascinating in science because, for example, right, what we have to detect, the maximal difference in time is 0.6 milliseconds for us, right? And for other animals with the smaller heads, the difference in time between the ears is even lower. So it was fascinated, right, how the brain can detect such small differences. The brain has to encode time, difference in time. 0.6 milliseconds, right? It's a smaller difference than the shape of an action potential in the brain. So it was a fascinating research, right? And this engineer proposed that if there are neurons that respond when receive coincident inputs from the ears, and they have different delay lines from the ears, maybe there are neurons that are responding to different time differences. And they could be, that's how our brain could be detecting and encoding interaural time differences. And actually research in owls found that the nucleus laminaris, the region that detects interaural time differences, is very consistent with the Jeffrey's model, right? It doesn't happen all across species, but it was actually found in owls that the Jeffrey's model of sound localization of detecting interaural time differences was found in the brain region that detects interaural time difference in owls. So it has to be, our research has been used to test models. And also, for example, these are models of human sound localization, how we localize sounds, right? We are processing information between our ears, detecting difference in time, different intensity, right, and different in frequency powers. And this is how we detect the location of sounds. And these models, right, they propose mathematical operations, right? Some time of averaging somewhere in the brain, cross-correlation somewhere in the brain, in our brain, mathematical operation, and even we detect difference in times and intensity, and they propose, we have to multiply interaural level and time differences, right, in our brain. Mathematical operations, right, which are important for modeling the brain. And actually, our research in owls actually found that these mathematical operations are performed by neurons in the owl. We found time averaging somewhere in the owl brain, right? Cross-correlation, neurons are doing cross-correlation, and actually a similar discovery we published in Science when I was at Caltech is that actually we found that neurons in these midbrain map neurons that implement a multiplication of interaural time and level difference, and these modelers, they send a message, thank you very much for finding that the brain can compute multiplications of interaural time and level difference, and when we published this in Science, they email us. So basically, the specialized sound localization of owls, right, have resulted in some mathematically elegant mechanisms in the brain of owls, right? So maybe that's a specialized sound localization behaviors, means a specialized brain function and mathematics, right? Elegant mathematics in the brain. All right. So specifically, the discovery of a map of auditory space in the midbrain suggested that there is a place code, right? This is the maps, right? It was an important discovery because there are maps in our brains, right? Brain maps are supposed to be for many things in our brain. However, this map of spatial location, it kind of suggested that this map by the location is a place code, right? The activity is encoding location, right? This was something that was suggested at the beginning. However, analysis of sound localization has shown specifically that owls underestimate peripheral location. There's a frontal bias for peripheral location, not just owls, all across species, including humans, right? We have a frontal bias for peripheral locations, and this is why we started realizing that this map of auditory space, a place code could not be able to completely implement the location of sounds, right? So we had to do some research to investigate actually how is this map auditory space encoding sound location. And with a dear colleague of mine, Brian Fisher of Seattle University, we were both postdocs at Caltech, we started thinking, there has to be something, how is midbrain map, right? Implementing this bias behavior, right? And what we start hypothesizing and finding, considering, is that yes, there is a map, but there are no uniform response properties across the map. For example, neurons in the front are more narrowly tuned, right? And there are more neurons tuned to the front than to the periphery. And we started thinking of a hypothesis, right? That maybe these non-uniform response properties, right? Maybe the shape of the tunings are encoding the reliability of the cues, right? And this pattern of response properties are encoding expectation, and maybe no place code anymore. And we said, well, maybe what we implemented, and my colleague mathematician found that maybe a global readout by a population vector readout of the map, if neurons, all these things are encoded, then maybe some statistical inference could be implemented by the brain for sound localization. So we had to test this hypothesis, right? Right, whether this encoding of reliability of interval time difference. This frontal bias was for horizontal positions. Owls use interval time difference for horizontal positions. That's why we had to investigate interval time difference coding, right? And then, so we had to investigate whether the reliability and expectation of interval time difference is represented in the brain, and that's why to test this hypothesis of statistical inference by the brain, we had to investigate things. And first thing, whether and how the reliability of interval time difference is encoded in the midbrain, right? Is a midbrain encoding statistics, right? So the reliability of interval time difference can be estimated by acoustic properties of the head, right? That's what it is. Because usually we are hearing sounds. Now you're just hearing me, but usually you hear sounds, right? That sometimes disrupt the cues, alter the cues of sound localization, and because acoustic properties of the head, right? There are different locations, there are different frequencies that have different variability, right? And reliability of cues. We did this analysis based on the acoustic properties of the head of owls, and what we found actually is that high frequencies are most reliable for frontal location, low frequencies more reliable for peripheral locations. And then we decided to test that. And a student, Fanny Cassettes, a person in the lab, she's now a faculty in France, recorded all across the map after we found these different frequencies are different reliable for different locations. As he actually found that across the map, neurons prefer different frequencies, and their preferred frequencies really matches this anticipated statistics. So basically, what we found is that in the owl brain, the brain predicts reliability of cues and predicts statistics, right? Honestly, something that some of colleagues here today also investigate whether the brain predicts things, right? And we're here in the hearing system, right? Predicts things, and we investigated this in owls, right? But it has been investigated in mice and in birds, right? How how we predict statistics and properties of sound, right? So this is so we found that basically in the whole brain, the statistics of this Q for sound localization is predicted by the brain. And what we had to investigate, because you remember the slide where I tell you the statistics, the reliability is encoded by the shape of the curves. So we had to investigate if that was true, right? And the reliability, right, of things could be anticipated or ongoing, right? Because this happens with the properties of our sensory. This happens in life, in natural stimulation. So the stimuli, right, have natural statistics, but they also have ongoing statistics. So anticipated statistics can be computed for the properties of the head, right? And so we computed, based on our analysis of acoustic properties, the anticipated reliability of sounds at different locations, and what we found, right, is that the reliability, right, was higher, less variability of interval time difference for sounds in the front than in the periphery, and it has this pattern, right, anticipated reliability and ongoing reliability, which is a different thing that brains have to detect, right, can be measured by changing noise level in the ears, and that can be computed, and it has this pattern, a different pattern by changing noise level, right? So binaural correlation is when we are adding noise, right? So 0 is completely uncorrelated sounds, maximal variability of the cues, right? And lower variability of the cues when sounds are very well correlated, right? And less noise. And when we analyze the shape of the tuning curves to these two things, we actually found that the tuning properties were very correlated with this ongoing and anticipated reliability in the owl, which was consistent with the hypothesis of statistical inference and coding of statistics by the brain. So this is one first thing we had to test. And then also we were hypothesizing that there's a global population vector readout that is implementing this statistical inference in the brain. So we had to test that. And then certain laboratory members, present here right now, they recorded all across the response properties of the map. And it was tested if response properties of the map by a global readout, population vector readout, could be implementing this biased behavior, and it was actually found. Right? When simultaneous recorded of different parts of the map by this population vector readout, it could be predicted the actual interval time difference of the stimulus. The estimated interval time difference was very close to the interval time difference of the stimulus. And for response properties of all across the midbrain map when noise level was changed, also, which in normal behavior induces stronger frontal bias when there's more noise, right? We also found that the population responses, a global readout of the map would also increase the frontal bias, right? Meaning that there are more neurons, more frontal neurons responding for noisy sounds, voting for the front, and therefore a population vector readout would also implement this frontal bias in this midbrain map. To continue testing our hypothesis, right? This was a recording of responses, population responses, but we said, well, can we test it with some behavioral testing to see if this is a global readout of this map, right? And then, because our discovery that across the map, neurons prefer different frequencies. They prefer high frequencies in the front and lower frequencies in the periphery. Then, if we are having owls localizing sounds and we present them sounds with different frequencies, right? For example, if we are presenting high frequencies, the sound, even if the sound is in the periphery, we're exciting neurons, maybe some frontal neurons that prefer high frequencies. So if this is a population vector readout, if frontal neurons are voting for the frontal bias should be higher for high frequencies. And we stimulate with sounds with low frequencies that prefer neurons in the periphery. If we have a sound in the periphery, the frontal bias in the periphery will be lower when stimulated with lower frequency sounds. If there's a global readout of the map, right, and responses of neurons across the map are voting and determining sound localization. And actually, this testing of behavior of localizing how owls localize sounds. Owls turn the head, they don't move the eye, they just turn the head to sounds. Localizing sounds of different frequencies was actually exactly found this. For high frequency sounds, the frontal bias was much higher than for low frequency sounds. So this was a behavioral testing that perhaps was a global readout of this map and not just a place code by this map, midbrain map, and in implementing the actual bias behavior, right? Then finally, for this testing is, so we are hypothesizing that this statistical coding, and as there's many research, our brains predict natural statistics, right? And it's our life experience determining our prediction of statistics. So we decided to investigate if life experience determines changes in these predicted statistics. And actually, Keanu Jadron, a former lab member, now a postdoc in Columbia University, thank you very much. What he planned is that if we change the shape of the face, the facial ruff, with a cat, we change the feathers, which has been done. The acoustic property of the head changes, and he did this analysis. This is our normal frequency, right? As I told you, higher frequency is more reliable in the front, lower frequency is more reliable in the periphery. But when the rough cut, higher frequencies are no longer reliable for frontal locations, right? So this is, he did facial ruff on baby owls, right? And then when they grow up, the recording of midbrain neurons. And what was found is that this is in normal conditions, right? Frontal neurons prefer high frequencies, peripheral neurons lower frequencies. However, after rough cut, the frontal neurons no longer prefer high frequencies, right? So there's a very strong difference, right? Experience-dependent difference in normal conditions, a correlation, but when the rough cut is removed, the high frequencies are no longer more reliable in the front. There's a change in the pattern of preferred frequencies by the brain. Another thing that we consider is baby owls. They still don't have a facial ruff, and it was recording in juvenile owls that also for the frontal neurons for juvenile owls, they also are responding to lower frequencies, right? So there was also a significant difference in juvenile owls that have different statistics, right, of these binaural cues, right? And the brain is responding and predicting statistics and responding to predicted statistics, right? This is something that happens in the brain, and there's several research that the brain ability of predicting statistics, right? And then finally, today is a comparison from owls to humans, right? And then. As I told you, the shape of our ears is different from the shape of the ears in owls. We have a Pena, owls don't have a Pena, right? And also the ranges, the frequency range, right? So the frequencies that owls can hear, right, is different, narrower than us. This is our range of hearing frequencies. So we can here different frequencies, right? Some humans. And also, in owls, there was a map, a midbrain map discovered in the external nucleus of the inferior colliculus, and also projects to the other map, the optic tecton, right? Contains another map. However, in mammals, there's no map of auditory space, right, in the external nucleus of the inferior colliculus. However, in mammals, there is a map of auditory space and visual space together in the superior colliculus, which is equivalent to the optic tecton in birds. These are the maps of auditory and visual locations. So there are differences in the maps, right, in owls and mammals. In addition, the cues that we use for sound localization, right? We use this frequency power different for detecting the elevation of sounds, and we use interval time and level difference for horizontal positions, and owls use interval level difference for elevation and interval time differences for horizontal positions, because they have this asymmetric here. So there are differences, right, on how owls and humans localize sounds. And in addition, our ability to detect interval time differences, our brain can detect interval time difference for frequencies up to 2,000 hertz, right? However, owls, specialized on localization, they can detect interval time difference for sounds up to 9,000 hertz. So, because to detect time difference, the brain has to encode the phase, the phase and the timing of the sounds, right? Owls the owl brain can detect the timing of sounds of very high frequencies, which we cannot, right? So there are differences in our different ability to for what frequency range we can detect interval time differences, right? Okay, and then what we decided to check, right, is our statistical inference that proposed for sound localization that we tested in owls. So is our brain doing something equivalent, right? Is our sound localization also depending on anticipated statistics, right, of this interval time difference, which we tested in owls. So a lab member, Rodrigo Pavao, who is now a faculty in Brazil, did an analysis of acoustic properties of interval time difference statistics in acoustic properties of our head, right? Like what we were doing for owls. And then, however, in human sound localization, it has been also done research, and the rate of change of interval time difference has been implied on how we have different discriminability at different locations of sounds. However, the analysis within the lab is a broader analysis of statistics, not just rate of change of interval time difference across locations, but also variability for perceiving multiple sounds, and then variability and rate of change combined together in this stat, right? Statistics, properties of interval time difference in humans. Just is our brain considering overall multiple statistics of this binaural cue. And actually, based on these statistics, we analyzed previous studies, and we found that in previous studies of sound localization and discriminability of interval time difference for different frequencies was maximally correlated with our statistics, the stat, right? This was done in free field, so we wonder, well, maybe this is not perfect because the human, if it sounds in different locations, different frequencies in free field, they're also using interval level differences, right? And we wanted to concentrate on interval time difference. So that we did analysis we are using earphones, just testing difference in time, how people detect the differences in time for different frequencies, and we also found, yes, that they were maximally correlated with this stat, overall statistics in the by the human brain, right? Indicated that, like in owls, our brain predicts statistics, and our discriminability is also dependent on these anticipated statistics. So this was testing of human sound localization, and with a dear colleague at Einstein, Elise Sussman, who also detects novelty detection in humans by electroencephalography recordings, specifically mismatch negativity, right? Is some signals that indicate detecting deviant sounds, deviant stimuli. We actually did EEG recordings in humans and investigated. We use standard and deviant sounds at different frequencies and different locations, and we found that the amplitude of this mismatch negativity amplitude changed for different deviants, and what we actually found that the amplitude of this mismatch negativity signal was correlated with the difference in interval time difference between standard and deviant, but the most perfect correlation when the statistics of the standard and the deviant sounds were included and generated the predicted correlation of the amplitude of mismatch negativity in humans, and specifically the statistics, anticipated statistics of the standard stimulus was determining these EEG responses in the brain. Right. So the brain, basically discriminability of sound location is dependent on anticipated statistics for human sound localization, right? And these brain oscillations, this mismatch negativity are also indicating this discriminability, right? We recently in owls also investigated something about brain oscillations. We investigated how the coding of salient sounds by the midbrain map neurons. We found for salient sounds, right? These lab members, right? For salient neurons that prefer a given location, when this sound is salient, the neurons, all the neurons that prefer this location are mostly correlated, and that gamma oscillations determine these correlated responses, right? So there is also, right, some electroencephalography and brain oscillations determining response properties in the owl brain. And gamma oscillations have been investigated in many brains and having like a role for attention, and that's how the brain encodes information and gamma oscillations. In addition to all our studies on the midbrain of owl, we decided to record the forebrain in owls. And a student that just recently graduated in our lab recorded the forebrain in owls, right? Recorded the midbrain map, and the region of the forebrain that receives projection from the midbrain map, that in the forebrain, it was found that the spatial tuning of neurons in the owl forebrain is much broader than the spatial tuning in the midbrain map, right? This also was found there's no map of auditory space in the forebrain, and neurons are more broadly tuned to space. And actually, it was found that they are receiving input from different parts of the map. So apparently, the forebrain even does a global readout of the midbrain map, right? And nevertheless, no map of auditory space in the forebrain. However, neurons are connected to different parts of they do a global readout of the midbrain map. They are more broadly tuned, but still their response properties change for sounds at different locations. This was found in owls, and actually in mammals, it was also found that forebrain neurons, for example, in the auditory cortex, are more broadly tuned to sound location, and there's no map, right? In mammals, in humans, there's no map of auditory space in the forebrain and in the auditory cortex. It was found, and even some research has shown that neurons in the forebrain respond to different patterns for sound at different locations, right? but nevertheless, there is no longer a map of auditory space in the forebrain. And only at least know that in the forebrain maps are a little bit broader all across sensory modalities than perhaps in the midbrain. So that is known. But this is the research we've done. And then our currently starting projects, we have fortunately collaboration with members for different parts of this country, right? And current and new lab members coming, Yongi. We are planning to continue investigating and concentrate on cellular synaptic and network architecture of this network, right? Just modeling neurons, right? And by doing in vitro recordings and electron microscopy, our collaborator, right? Mark Ellisman, electron microscopy in San Diego, right? We are doing electron microscopy in all brains to really determine the fine properties, right? The microscopic properties of the owl brain, right? And that's what we are planning to do, fortunately, and starting in our lab, right? And even doing behavioral recordings, right? That just continued our behavioral recordings in owls. So, conclusions, right? Hearing, essential brain function of hearing. We use to communicate, detect environment, even enjoy life, right? And some species like barn owls to detect sounds and find food, right? There is an optimal representation of acoustic natural statistics by life experience, which happens all across sensory modalities. Our brains predict statistics and predict things. It has done a lot of research for hearing, but also for visual detection and everything. Our brain predicts statistics. That for hearing, that actually the brain is implementing mathematical operations, right? Mathematical operations, our brain implements mathematics, right? So mathematics are actually in nature, right? Mathematics are important to describe nature, and our brains are implementing mathematics to predict nature and implement the computation of information about nature. And they are consistent things across owls and humans, right? But there are important differences of ability to localize sounds between owls and humans, right? So, well, then, thank you very much. Hope if you have any question or something, let me know. Yeah, thank you for being here. Yeah.
We have some time for some questions. Yeah, right there.
Very interesting talk. So,
thank you.
I was wondering, can animals also just rotate their heads to make a difference between the 2 ears in terms of the level and use that to...
Very interesting question. Yes.
On the flip side, if the owls have their head rotated, do they need to correct for that?
Yes, owls rotate the head because owls cannot move the eyes. So we tend to rotate a little bit the head, but also we move our eyes for sound localization. Yeah, we are even in, for example, right, to following conversations in a party when it's very loud, right? We can follow what somebody is telling us when it's very loud based on the direction of the sound, right? Sometimes we turn the head. It's true, right? And we usually we put the sound we are trying to hear. We just put it frontally in our. We tend to put it frontal in our heads, right? That's usually what happens. We use a lot of vision and also actually we hear, and sometimes So sounds are pressure waves, right? What sometimes happens is that sometimes with our bones, if the sounds are very loud and they are low frequency, we just hear it by vibration in our head, right? So that also happens in our brain, that detects movement of different bones, right, by sounds, and it also happens with some kind of hearing dysfunctions or something. Sometimes the bone, we detect the location of sounds, etc., but how the position of our bones that are touched by these pressure waves of sounds that happens. And sometimes we can dance in, if we don't hear, if we are deaf, sometimes we can hear sounds by vibrations in our bones, right? And very, very loud music, low frequency music, we can dance even if we don't hear it, right? Because our bones can detect also pressure waves, yeah. That's, yeah.
Anyway, I had a question about, you're comparing owls mostly to humans or mammals. I'm wondering about among birds, like, for example, birds of prey versus birds that are kind of more prey and they get worms, or, you know, like the differences in hearing. The head turning is interesting too, like why, you know, again, I guess they can't turn their eyes, but it's just a strange evolution that owls turn their heads
Yes.
when other birds of prey even don't really do the same thing.
Yes, honestly, most bird preys, they use vision to catch prey, right? And even some owls. So this is why owls, these are barn owls. That's another interesting thing. Owls, these barn owls are very nocturnal. That's how to localize food. They do it at night, and they localize food, mostly mice, a little at night by the sound. For example, it has been shown that when they are breeding, the male owl has to bring a lot of food for the wife and the babies, and they sometimes have to catch so many animals at night that they are very efficient sound localization. However, right, great horned owls are more diurnal, and they use a lot of vision instead of hearing, right? These polar owls in the Arctics, they catch prey in the ice or something. They use mostly vision. So yes, across birds, there's different function. And birds, singing in birds is very important. Hearing is very important for birds singing, right? Oh, yeah. Sarah Wool is doing this important research of the singing and predictions of the properties of songs and everything, right? Which could be related, right? We from different races, babies even could have preferences for different languages, different words, right? Because of our different races, we have predicted different kind of talking or something, right? So hearing is very important for birds for singing. However, sound localization, that's why we're talking about owls. Barn owls are very specialized in sound localization. However, animals, dogs, there are many animals that localize sounds in different ways, but they move their ears, right? And in different ways, right? So yes, there certainly differences across birds in sound localization too, not just between mammals and birds. Yeah.
Question back there, and then there.
Thank you.
Thank you so much for the talk. So I have two quick questions. So first is, does the vestibular system interact with the map somehow, like shifts it, or? The second question is, does the owl also show like some microhead saccade, like, you know, same as?
Very interesting. I was planning the vestibular system detects balance, position, right? So our ears, I don't have time today, but our ears also detect balance. The vestibular system detects balance, and also in owls, the shape, right? Let me just go to the ear shape. The vestibular. Well, I was going to the first. You see, our vestibular system, right? Semicircular canal, detect balance, position, right? But also in birds, there's some kind of detection of balance. vestibular systems, right? And also, it has been proposed that even insects, right, vestibular system also has happened, right, in insects somehow, but the antennas or something that detects, right. So across auditory system, the auditory system detects, they have vestibular systems that detect balance position in different ways, all across species, but yeah, in humans, in us, and mammals, and birds also. It's also detected, and it's very important for flying in birds and all that, right? For birds, it's very important. Yeah. So yes, yeah, no. Yeah, do side.
Hi, thank you for the beautiful talk. I guess I was curious how you think about how sound localization interacts with other behaviors or other tasks that we use our auditory system for, particularly because it seems like such a specialized mechanism that maybe evolutionarily developed very early on. And so how does that then interact with some of the other auditory processes that we have, which we think of as being much more like cortical in nature?
Yes. There is some research about it, the cortical, the cortex. That's why we want to investigate the forebrain, right? And specifically, well, you don't have time to talk about something important is for illusion. There's sometimes illusional detection of illusions, right? When sounds are confusing, sometimes we have illusions, right, of sounds at different locations. And what actually determines illusions, right, by the forebrain, or the perception, actual perception, is something that there is some role, right? It has to be, and so there are sound localization is it's a complex, it's a computation, right? There are other things that brains have to compute, right, in sensory systems, right? For example, in bats that do the distance of echolocation, right? Also, they have to compute echoes, right, between their vocalizations and how long it takes to come back the sound, right? So there's a lot of computations, and for us, sound localization is very important. Actually, as I was telling you, we follow conversations. Even to follow conversation in a party, we are localized. We are determining the direction of sounds. When we are walking around, right, in life on the street and everything, we just hear where trucks are coming or something. So sound localization is an important function. But hearing overall, right, there are many functions about hearing, right? It's to communicate, right, communicate with us. Also, to hear music, right? It's a pleasant life, but also just detecting nature and something. Yeah. There are differences. Yeah, that's what it is. Yeah, it's a future research of actually complete understanding of hearing still. Yeah.
Okay, we had two more questions called out here, and then if you have further questions, you can meet us out in the lobby. First, you.
Very nice talk.
Thank you.
Very nice talk. So there's something that I didn't understand is that, so the more reliable responses are the high frequencies to the front, the lower frequencies are more reliable on the side. But when you do the task, when the barn all, you give it a sound, they move their head, they move it to the front. Is that always the case for low frequencies or for high frequencies? Because if they are looking for reliability, maybe they should look the other way for lower frequencies.
Yes, exactly. Reliability will change its biased responses, right? However, owls can localize sounds, right? Well, the issue is that, honestly, I don't want to talk about ambiguous signals today because it's a complicated thing. For single frequencies, right? For single frequencies with earphones alone, the signals are ambiguous, and sometimes owls can think the sound is here or here or here, and they sometimes turn their head to different locations. That also happens with us when we have earphones at the different frequencies, because the single frequencies have periodic signals, and they are ambiguous. So that happens, right? This is when in free field stimulation, we sometimes use interval time difference, interval level difference, and that disambiguates the location of sound, and even owls use for sound localization different cues, and they disambiguate. So this is what happens. So there is an overall, so owls localizing sounds, yes, the reliability changes across frequencies and locations, right? However, right, this is how the brain predicts reliability of different sounds. However, right, sound localization can be performed for different frequencies, even for owls at different locations, right, based on these anticipated statistics. And this is why our brain predicts statistics, right? To just perform, right, sound localization efficiently for any sound, right? We have also difficulty, right? Right? Localizing sounds for, well, as I told you, well, the issue is that why our brain, right, we can detect difference in times for low frequencies. However, difference in intensity for high frequencies, and that's how we use interval time difference and interval level difference for detecting the horizontal position of sounds. And then the frequency changes at different elevation. That's why, yeah, it varies. That's what will happen across location and frequencies. We are implementing sound localization. Sometimes there's a little bit difficulty, right? For example, for low frequencies, the difference in intensity is lower because the low frequency goes through walls and head, or that's one issue. And sometimes there are phantom signals, right? Phantom perception of phantoms. That's what happens, and we want to investigate that.